Um, let me see.
Yeah, it is coming. Good.
Okay.
Yeah.
Perfect, perfect, perfect.
Very nice.
So first of all, let me thank you, Poonam and Alejandro, for inviting me to be here in this very nice symposium. This was a great idea in this kind of problems, the commitments of you share these works and all these collaborations with people from India and Brazil have been a very great idea, and we look forward to continue money is a very important issue. We can try to continue our collaboration because it seems that the area in which we were working, that are retrovirals, it seems that we have a lot of work to do, and that work could be very useful for both of our countries that have very similar problems configuration. So I will go to my talk after. Thank you for inviting me here. Trying to continue what Alejandro was talking about, I will small molecule with pharmaceutical properties in which we use it as a model for proving all these crystal forms, trying to get all these crystal forms that Alejandro was talking about. And as Alejandro and Beatriz were talking, these solid forms have different pharmaceutical as well as chemical and physical properties, and this can be modulated in such a way and the biological activity of this molecule, and also to repurpose some of the medicines that, for example, the one that I have been doing now for COVID-19, this pandemic problem. As a model, I will show you one molecule that's very small. This one is called 5-fluorouracil. This 5-fluorouracil is very cancer drugs that have been using for a long time. He's got problems like variable viability, and even when they have very several functional groups, had to be given intravenous, causing discomfort in the patient and risk of contamination, as Alejandro was saying. And also the main problem is that they have low specificity with so that's combined with the short half-life of about 30 minutes was resolved by using another similar compound that is the 5-fluorocytosine. This 5-fluorocytosine is as changing the oxygen in here by the NH2 group. In this case, this fluorocytosine was previously used as an antifungal compound, and using some, it can be used to produce this fluorescein block as gene therapy. So that can be directed to the site of the tumor and inviting side and problems that can be dangerous for us and the other one. So we use this cytosine as a molecule, as a starting molecule, as a model for our works in solid state. This work from a couple of my students, and we have been working in this area for at least 10 years. That said, I can say you that these have several solid forms, mainly hydrate, as you can see here, all of them with different amount of water. For example, this monohydrate in here with one API and one water molecule. Here with 2 API and 5 water molecule. However, the one that is used in, as pharmaceutical industry is this one. That is the anhydrous phase with no water. That's because of the stability and physical properties of these solid forms. This 5-fluorocytosine the biopharmaceutical classification system is called class 2 with high permeability but low solubility. It has some problems with stability. The main solid form used in the pharmaceutical industry to produce all the medicines, as I told you, is the anhydrous phase, and this anhydrous phase have problems with the hydratation. If you put this solid form in a relatively high humidity environment, it will turn into the monohydrate form. We're trying to work with this monohydrate form for a long time. What we discovered was that from this monohydrate that was produced by the hydration of the anhydrate form, you cannot get back to the previous form. Even with the heat treatment, what you recover is a known phase that is not reported, but you cannot get back to the anhydrous phase previously used by the pharmaceutical industry. So this is very bad for the pharmaceutical industry, since you can you have some stability problem with your main products. So we move on trying to use what we call crystal engineering to produce that forms. As Alejandro said, there are a very high, very wide universe of different solid forms that goes from hydrate, unsolvate, salt, Cocrystals, all these ligand spaces where you can get cocrystal hydrated or ionic cocrystal or salt hydrated, and that gives you a huge universe of different solid forms that you can get. So using this molecule as a model, we decided to study these new forms, trying to see how well you can perform, how we can try to get more of these solid forms. That said, it's important to point out here that these new solid forms have been treated very seriously by the agencies like the FDA. The FDA has given regulatory classifications guide in 2019, 2098 about solid form, especially cocrystals, given the importance of this in pharmaceuticals. So we decided to apply crystal engineering, trying to see mainly focusing on the symptoms and trying to see if we could model these symptoms, trying to work with them, use it to design new solid forms. So the main tool in here is for the rational design, is the use of the Cambridge structural database, using this tool as analysis tool for analyzing the symptoms. That means how is the relative probability of the different intermolecular interaction that can be produced in order to use that probability as a key tool to the design of new materials, new solid foreign physicochemical as well as biological properties. That means redirecting intermolecular interaction like hydrogen bonds, charged acidic hydrogen bond, fire interaction, and so on. In the specific case of this 5FC, we have mainly three different types of intermolecular interactions that forms dimer. This one NHO with three different dimers forms, being this one called form 3, the main one with higher probability, that is the NHN with double NHN in here. So to use that as a tool for design new universe of these solid forms, working mainly with salt, cocrystal, also moving to a more interesting area that could be the drug cocrystal. So let me explain a little bit about that. One of these means the point here was trying to synthesize a very bright white area of different solid form, try to control this acidity of this solid form. So using the different in delta pKa, we can control if we can produce salt and cocrystal. For that, started with some carboxylic acid, mono and dicarboxylic acid, using this carboxylic acid as a main co-former for this crystallization. Using that, we saw that with delta pKa bigger than 2, we normally used to have salts. With delta pKa less than 0, we usually get cocrystal. However, in the middle between 2 and 0, we have a area called continuum in the form, and the solid form that you get, it depends on what is the main compound that you have. In here, in the case of this 5FC, we have mainly salt formation. So we try to study how these different intermolecular interactions forms take place, and how this mold the different forms that we have. During that, we were able to move from one point to in, and we saw that the main difference between salt and Cocrystal is that in the case of Cocrystal, you have mainly homosymptoms forms in your solid area. You have mainly heterosymptoms forming. In the case of Cocrystal, as you can see, we have our NHN, NHN dimer, as well as the NHO, NHO dimer. In the case of the you have mainly heterosymptoms, where this 5-FCs mainly were interacting with the carboxylic acid in a way that it was not doing in the Cocrystal. We're trying to see where exactly you go from Cocrystal to salt, and we were able to do that with malic acid, in which we get a structure that was partially Cocrystal, partially salt, in terms of the intermolecular interaction. Most characteristic from Cocrystal and salt, in which from one hand you have these heterosymptoms with the FC were interacting with the carboxylic acid, and on the other hand you also have these interacting homosymptoms between 5-FC compound. Working also in the Cocrystal area, we were able also to go to the opposite extreme and working to the area of what we call a big different crystal form by itself, and had been getting a lot of attention, mainly from the patents point of view, the intellectual property point of view, since that can be make a solid forms. So we put a lot of effort trying to get this solid form, and finally we use it picric acid. This picric acid was very a nice conformer with 3 carboxylic groups with 3 NO2 groups and 1 oxygen. So a lot of interactions able to form. And here you can see 1 FC is protonated and 1 is not ionic Cocrystal. Moving a little bit further, we move trying to design now Cocrystal with other drugs that are drug Cocrystal. In this case, the problems are a little bit hard because you have to combine in the same crystal 2 molecules of different APIs. But these APIs not just to be complementary from the intermolecular interaction point of view, but also the pharmaceutical and medicinal point of view. So we do that, combining this 5-FC with the 5-fluorocytosine, that is the drugs for treatment, with caffeine, and with also with isoniazid. Isoniazid is used for tuberculosis treatments. That's one of the main respiratory issues. Now let me go one by one. The case of the combination of the 5FC with 5FU, remember that 5FC is a prodrug of the 5FU, so you can have different forms of working. The 5FU will work mainly from the beginning, and after that you can use genetic therapy to produce a little bit more of 5FU, starting from 5FC. From the solid form point of view, what we saw was that even when you have this cocrystal, the cocrystal is formed by dimers of 5F2 and 5FC. So even when you have this this combination forms, they retain this own almost symptoms. And this is very important things, because you can now try to do what Alejandro was talking about, that is this solid solution, trying to mix more different amounts of these two molecules, trying to see if you can get, for example, 2 to 1 or 2 to 3 combination. Now we are working on that area as a result. This can be done also from the cocrystal point of the same intermolecular interaction. These are isostructural. So this is an area where these solid state solutions can be used in a very nice form. Other area that we used it was the caffeine. The caffeine, as you can see here, it crystallized with 2 molecules of caffeine and 2 molecules of 5FC. This crystallization retain the 5FC 5-cytosine homodimer. So even when the caffeine were able to interact with the 5FC, it was not strong enough to compete with this homodimer. That's a very important issue that we use it here as a conclusion. This caffeine, as well as other cocrystals that we produce, have very that is similar. This solubility study showed that even when we have a cocrystal, you can have similar solubility, aqueous solubility for this compound. So you have here a drug crystal with different coformer, and this solubility shows that the solubility has been improved. What is very important, since when you have cocrystal, trying to avoid the change in the biological parameters. What is more important with these new solid forms is that the water uptaking was prevented. So these new forms have a very high stability. We were able to do some long-term study. The solid forms remain stable for more than 2 years in the same solid form without uptaking the water molecules. So that's a good point, because you have this a good solubility, and you have also no hydration. So this allowed us to produce some patents. That's very important issue, not just to remain with the science, but also try to get to the the pharmaceutical industry. I convinced them that these new solid forms can be a tool to resolve some of the problems that our country have with the drugs and trying to get better drugs using new solid forms. Other that mean drug Cocrystal with this molecule in here, this Cocrystal is 5-FC here with isoniazid. Isoniazid, as I told you, is one of the main drugs used for the treatment of tuberculosis. And so this combination of drugs was mainly done thinking about lung cancer. That is very common effect after the tuberculosis. So having both diseases at the same time, tuberculosis, cancer after the tuberculosis, as well as fungal infection in the lung, have been very problematic issues. So this drug was designed for this treatment, for the treatment of this concomitant diseases. We were able to visualize that for solvent, as well as the mechanochemistry procedures using what is called green chemistry. This mechanochemistry way synthesis is very important for the android, since it's more easily to escalate, since you use SAS, a small amount of solvent is what is called green chemistry, and it's very attractive for the pharmaceutical industry today. So this, the solubility profile of this is similar, even when these two, the isoniazid and the 5-FC, similar, different in solubilities. This Cocrystal have a solubility similar to the 5-FC. So even when you have, we have here the drug Cocrystal, we don't miss too much of this solubility. Is a good to the market. Again, it was possible to enter with a patent in here, what is very important to mainly to show our science agencies that we can have a product as a result of our research. Moving a little bit further, we're also trying to get a little bit more information about. To do so, you have to go to the what's called charge density analysis. That means that in order to see how these interactions occurs, how these changes in the intermolecular interactions are result of the star density, the main work here was trying to study experimentally and theoretically the charge density of these compounds. It was done by high resolution X-ray diffraction analysis, combined with three-dimensional periodic quantum mechanical calculations, and also a combination of both, what is called X-ray constrained wave function, as a way to get this information. This theoretical can be done in gas phase periodic. We mainly combine the three of them, and the combination with the charge, experimental charge density was done through the crystalline theoretical calculation. In the case of experimental charge density analysis, they use what is called multipolar model, in which the density of the atoms are divided in the density of the core, spherical density of the valence shell, and the deformation density that use some parameters to study how this valence shell is deformed for each one of the atoms. The problem here is that you have a very high increase in the number of parameters, so you have to go to very high resolution in order to do that. Let's say normally you go to a normal X-ray study, you go until 0.7 angstroms. Now you have to go to 0.4 or 0.3, something like that. This multipolar analysis use a real spherical harmonic with octupoles, meaning S and P orbital for carbon and nitrogen, and hexadecapole for metallics or things more heavy like sulfur or chlorine, for example. And the radial function associated types, for example, in here. This allow us to study the molecular topology using quantum theory of atomic molecule. That means that you can have a very higher quality information of the charge density, and you know where and how to divide, how to separate the different charge density from the different atoms using what is called scalar field. The gradient of the density show this information, where the is 0 shows what is called the bad things, that is the volume that's of the charge density that belong to specific atoms. The Hessians of the charge density, that means the second derivative, will show the critical points. The diagonal of this is called the Laplacians. The Asian value for this will the positions of what they call points, and that show the nuclear critical points for where the atoms are, at the bond critical points that are in the middle of the intermolecular interactions, as well as the bond, covalent bond. As you see here, you have this critical point will give information about the ellipticity of these bonds, meaning how pi or how sigma or how concentrated polar is your interaction. And this gives you what is called molecular graph critical point that show you how this polar here, you have the carbons, the nitrogen, the oxygen, and the critical point here in red that show you where the density start for one atom and two other, and show you, for example, here the polarity, the bigger here the oxygen, the smaller here for the carbon, and again here the carbon closer to the carbon, not to the. This also allow you to get the oxidative potential, which is very important because is how your pharmaceutical molecule will interact with the ligands, or with how the ligand will interact with the proteins site. So this information can be also get by DFT, circumsystem field relations, using a model. For example, B3LYP, combine it later in Gaussian function, and this allows you to get the information. When we apply that to our drug Cocrystal, allow us to get this fields in here. These are the vaccines. Here we have the molecule in here with all the atoms, all how the density of the molecule is divided into the density of different atoms. These are the volume, call it the vaccines, that is how these density are divided. So that gives you information about how this molecule interacts. Here you have, for example, the contour of the Laplacian of this. They give you the concentration of the charge density. For example, here is the information of the intermolecular symptoms between these two molecules. The nitrogen, put it to the hydrogen atoms on the NH2 or the 5FC, and also in here the formation of the lone pair of the other nitrogen of the 5FC, pointing to the nitrogen, to the hydrogen of the so this gives you more information about this charge density, very clear information, how these molecules interact. Here you can see a little bit further information about these symptoms. Here you have to see this critical point. Comparing this information here in the bond critical point, you can have information about how important these interactions are, how strong are these interactions. Combine this with the potentials molecule will have in biological site. For example, here you can see how this density of the hydrogen finishing here, how this nitrogen one starting here, how that these are can be divided in this formation. This is from the experimental experiment showing the electrostatic potential in here, the more hydrophobic regions and the hydrophilic region, and how these two regions are combined. This is very important because this is the phase, this is the form, how these molecules will interact with the active site of the proteins. This is crucial information when you want to go to the docking problems and trying to make a more advanced drug design and trying to understand how these molecules interact with your active site. Okay, let me finish with that. Just show you a little bit of my group that are mainly the ones that have been doing most of the work. So our collaboration, Dr. Plantando, and also Alejandro Ayala from the Federal University of Ceará, as well as our collaborator in Germany and Mexico as well. And of course, the people that pay for our works, and to you for your time. Hello.
Yeah. Thank you very much, Professor Javier, for very nice talk. Are there some questions? Please drop the message. I have one question. Venu is saying he has some questions. So I will unmute Venu. 1 minute. Yeah, unmute. Venu, now, Professor Venu, please.
Yeah. Thank you, Professor Poonam. Thank you, Professor Ellena. That was a very interesting talk. Even presentation slides were quite clear to go through them as you are doing presentation. I have a couple of questions. One of them is you were talking about the pKa rule. Generally, we consider that pKa rule, if it is 3 and above acid-base difference is there, then you would expect to see salt. In your case, it is about 2 pKa differences, 2 and above maybe, and then you have seen salts. Is that very common or something to do with these systems only?
It's not very common. This area where the start, where the co-crystals start, is a very fuzzy area, and it depends of specifically of the compound, and it's clear that higher than 3 delta pKa you will have a salt. Before below 0 you have a co-crystal. In the middle you don't know, and because that will depend of the intermolecular interaction and how this competition will go. Competition between your homosymptoms in co-crystal and the hetero symptoms in the salt. So it will depend on how the different forces, how this different small difference in the charge density will vary from one to another. So in this case, the homosymptoms will allow us to, it will break very easily, mainly with carboxylate. And that will stop at 0.1 delta pKa. After that, you have a salt. But other cases, you will have to study specifically for that compound. That's not a result that you other compounds.
Okay, I have another question. You were talking about a co-crystal with the picric acid. You say that
Yes.
you say that as a ionic co-crystal. We know that Dario Braga introduced ionic co-crystals and all. I'm wondering to see, you also introduced FDA guidelines and what should be allowed by them. According to you, whether this should be called better as salt or it is a ionic co-crystal. What's your opinion on that?
These solid forms from the last 10 years. So this lot of grammatical problems and different definitions. I guess that it will depend in which area you are working.
Venu, he is asking in which area you are working.
No, I work in the crystal engineering area and also pharmaceutical co-crystals. So
for a scientific point of view, it's easy to get into a decision.
For example, this system is put forth this is not a picric
ionic
acid. Is not
crystal,
a.
but the agencies of the patent law, that's a complete error, and they just don't care about science, and that they make their own that sometimes have nothing to do with science. And so I guess that if you are trying to get a new chemical entity, to get a new product in the market, you will, if you are just from the scientific point of view and you want to just important for you with the science, well, that's another definition that you have to use. So say, as I told you, use to look at it.
Yeah, thank you. I understand. I have last quick question, which is the solubility advantage that you entered for the 5-fluoro-cytosine and so on co-crystals. So what you presented was relative solubility enhancement between by changing the coformers, and you got the several of complexes. What I am trying to ask, what I am trying to check is if you take 5-fluorocytosine alone, and upon making the complex, it could be with any of the co-crystal coformer. What is the maximum amount of solubility enhancement you have seen?
Yes, we mainly the prodrug is very high, have a very high solubility. So that's not a problem for that. We didn't want it to change it. What we wanted to see, we don't want to lose a lot of this solubility when you get the co-crystal. You understand? Okay, you can get a co-crystal, but if this co-crystal is not soluble at all, that will not be useful. So the new co-crystal that was previously presented by the fluorocytosine. So that was the main point, not to improve the fluorocytosine solubility, that it's high enough. Avoid to lose that when you get the co-crystal. That's the main point, trying to see to prevent the loss of the quality that you have when you started.
Okay, thank you. I leave Professor Poonam to have any questions on this charge densities. Then thank you very much.
We can talk later.
Thank you, Javier. Maybe with if there are more questions, people can contact you individually, because we are now running short of time.
You have my contacts.